We have a really outstanding lineup for the afternoon session today. Starting things off is Professor Justin Sonnenberg. So Justin Sonnenberg, he's an associate professor in the Department of Microbiology and Immunology at Stanford University School of Medicine, where he studies the gut microbiota in health and disease. In 2009, he was the recipient of the NIH Director's New Innovator Award, and in 2011, he received the Burroughs Wellcome Fund Investigators in Pathogenesis of Infectious Disease Award. He and his wife Erica are co-authors of the book, The Good Gut, Taking Control of Your Weight, Your Mood, and Your Long-Term Health. Professor Sonnenberg is one of the country's top experts on the microbiome, so please join me in welcoming Justin Sonnenberg.
Okay, it's great to be here. Thanks to the organizers for putting this meeting together. Thanks to Akil for the invitation. So I've studied the gut microbiota for about the past 15 years. I started in the field before most people had heard of the gut microbiota or microbiome, and I've watched this field really blossom to kind of insane levels over the past several years. And 8 years ago, I moved to Stanford and started my own lab there with my wife. And now we run a lab that's focused around basic science. We're actually biochemists, molecular biologists by training. But over the course of our research, we really started to notice that we were living our lives differently, eating differently because of this research that we were conducting. And so I've been going to more meetings like this, talking to people who care about food, who are interested in health from a very different perspective than the biomedical community from which I come. So anyway, it's great to be here. I notice these types of meetings also always have a lot better food than the meetings I'm used to going to. So anyway, it's great to be here. So I thought I would start off with this, what I consider to be the most beautiful image of the gut microbiota taken to date. This actually was taken by a graduate student, former graduate student in my lab, Kristen Earle, and it actually is an award-winning photo. She entered it in the Nikon Small Worlds contest, a photomicroscopy contest. It took second place in that last year, so we're very proud of this image. So what this shows is it's actually a mouse colon harboring a human microbiota. So that mouse colon's taken out of the mouse and then sectioned, and then we stain it with fluorescent probes and image it under a microscope. So you're seeing that cross-section here. And so down here in the bottom right corner, this is the host colonic epithelium. So each one of those blue circles is a nucleus of an epithelial cell. And then in the top left, up here, these are all the gut microbes. Every one of those little balls and rods that's fluorescently colored there is a different bacterial cell. So you can see the incredible density of this microbial community and how close these microbes are to our host tissue. These are the microbes that we're harboring in our colon. And then there's this this green layer running in between here. Anybody want to take a guess at what that is? Mucus. Mucus. Mucus is always green. You learned that as a kid. Kristen she was very sensible when she was conducting this project and always made sure the mucus was green. Okay, so we harbor trillions of microbial cells. There's been a recent paper adjusting these numbers relative to our human cells. Frankly, it doesn't really matter how numerous these cells are. What is clear is that they're having just this huge impact on our biology. They have a collective genome that's just amazing. When you consider that we have around 20,000 genes in our human genome, they have over 100 times more genes in their collective genome. So they're bringing metabolic capabilities and developmental cues to us that we haven't evolved or haven't needed to evolve on our own because we have these great microbes. And so the field is moving very rapidly from trying to understand who is there, census taking. Many of you have heard probably of the Human Microbiome Project. And now science is transitioning into actually what my lab is specialized in, which is what are these microbes doing? Can we understand the mechanisms by which they operate, how they respond to perturbations? And we know that one of the major things that they do is they degrade dietary fiber. This is what they live on, and I'll talk a lot about this today. But what is clear is that we actually, you know, we can't define ourselves simply as a collection of human cells. We have to define ourselves as an ecosystem, that we actually are a collection of human cells and microbial cells that come together to work in concert. And it's a really integrated, complex system that we still don't really understand that well. Okay, so it's been well understood for many years that the gut microbiota impacts our digestion, it impacts gut health, but what has been amazing over the past 10 years or so is to find out that these microbes, although they're confined within our gut, actually reach out into the rest of our bodies, biochemically speaking. So these microbes actually influence our immune system in many different ways. How likely you are to get a respiratory infection or how you respond to a vaccination is dictated by your gut microbes. These microbes also are in control of our metabolism, so there's a connection to how we harvest energy from our diet, how we store it. I'll show you some data related to obesity that's really interesting. And then a variety of other processes in health and disease can be influenced by these microbes, including them sending signals to our central nervous system, potentially influencing moods, behavior, neurodegenerative disease. So there's no system in our body that goes untouched by these microbes in some direct or indirect way. Okay, so the gut microbiota is really a control center for human biology. Okay, so I wanted to start off with a little data because I think this is a nice way to vividly illustrate the impact the microbiota can have on our health. So back in 2005, Ruth Lay, she was a postdoc at the same time I was a postdoc in Jeffrey Gordon's lab at Washington University. Jeff is one of the pioneers of this field of microbiota research. And she conducted a really nice experiment where she just looked at the composition of the gut microbiota in mice that were lean or genetically obese. And she sees these two major groups of bacteria, the Firmicutes and Bacteroidetes, at about 60-40 in the lean, and it shifts in the obese. And so she thought, well, that's interesting. Let me go look at some obese humans. And she saw a similar trend. It's not exactly the same, but skewed towards Firmicutes, and lean has a more normalized ratio, more even between the two groups of bacteria. When you put obese individuals on a diet, whether it's carb restricted or fat restricted, as they lose weight, the ratio normalizes. So what's going on here? How is the microbiota connected to obesity? Is it downstream? Is it a result of obesity, a result of how lean you are, or is it actually contributing to and potentiating obesity? And how do you get to this when you're talking about a complex ecosystem? Some of you may have heard of Koch's postulates, how if you want to find out if a bacteria is causing disease, you isolate it from a diseased individual, you introduce it, and see if it causes disease. If you can get rid of it and cure the disease, that's better. Barry Marshall won the Nobel Prize for this with Helicobacter pylori. But that's kind of the standard for determining if a microbe is causative for something. But how do you do that when you're talking about an ecosystem that consists of hundreds of species of bacteria? Well, they performed a very clever experiment. They took the normal microbiota from wild-type mice, and they put that into mice that are known as germ-free, or mice that lack a microbiota. They live in sterile bubbles. They can introduce the microbiota of their choice. So these normal mice received a normal microbiota. These normal germ-free mice received the obese microbiota, the skewed microbiota. And they asked the question of, if these groups of mice eating the same exact diet have different microbiotas, how does that impact their weight? And guess what? The guys that received the obese microbiota gained weight more quickly. So that means the skewed microbiota was actually somehow causing or potentiating obesity in this instance. And that was really the first vivid demonstration that you don't just have a linkage between the microbiota and health, but you can have phenotypes, physiological phenotypes conferred by the microbiota. Now, today I'll be showing you a mix of mouse data and human data. I'll always try to show you what's going on in the human realm. Those studies are harder to conduct, but they give some idea as to whether we're grounded in what our conclusions are from the mouse model data. So here's an example of trying to do that same experiment in humans, taking obese humans, giving them microbiota from lean individuals, and seeing how that affects their metabolic markers. And the title says it all here. Transfer of intestinal microbiota from lean donors increases insulin sensitivity in individuals with metabolic syndrome. So here they're actually now it's a transient effect, but the recipients didn't change their diet. And so the thinking is that their microbiota reverted over time because they weren't reinforcing this new lean microbiota that they received. So a major question in the field has been, to what extent can we manipulate our own biology via the microbiota? And the idea isn't to sever these really important, highly evolved connections, but to create a set of tools and a knowledge base that allow us to manipulate these microbes that are in turn manipulating us, to put the reins back in our hands. And there are a variety of strategies for doing this. Many of you have probably heard of fecal microbiota transplants for Clostridium difficile colitis. This is one of the more exceptional demonstrations of how the microbiota can confer health in the human situation. But there are a variety of other approaches, ranging all the way down to engineering microbes that can do special things, conduct tasks in your gut, marrying the ideas of synthetic biology to the gut microbiota. But with all of these strategies, I think even put together, there's not really any evidence that any of them will be more powerful than diet in modulating this community and dictating how it can change our health. And so I wanted to give you just a thumbnail sketch of what metabolism is like in our gut microbiota. So we have dietary fiber, or what we like to call microbiota accessible carbohydrates, or MACs, that pour into the system. These are the major fuel for gut microbes. They get fermented by primary fermenters, converted into short-chain fatty acids and other fermentation end products. Those can be absorbed into our system. They serve as a salvageable form of calories that would go unrealized in the absence of a microbiota, but also they play important regulatory roles that we'll come back to. There, of course, is intense competition for these resources in this really dense environment. There are other microbes that actually feed off of the fermentation end products, and that's what drives their metabolism. So this is really a complex food web. It's an ecosystem. There's predator-prey interactions with phage occurring, and so we have to remember that everything's connected in there, and it's all influenced, and there are ripples from what we put into the system, what we're eating. Okay, so let me switch over to the Western diet. So I mentioned dietary fiber is important for feeding the microbiota. Most of us don't eat a lot of dietary fiber. Western diseases are on the rise. We all know that autoimmune diseases are trending upward. Same with obesity. If you look at projections, they all go up. And the question is, can we imagine a future where that's not happening? You know, I watch a huge addition going on to Stanford's hospital right now, and I think of the hundreds of people that are going to occupy those beds and what diseases they're going to have, what diseases most of you people treat in your daily lives if you're in healthcare. And these diseases are preventable, and I think it probably has something to do with the microbiota. Can we imagine a future that looks like this, and how do we do that? So if we picture Western diseases as this tree, we have a very reactive approach now, right? We're talking about treating symptoms, developing drugs. Most of my colleagues in the biomedical field are studying disease mechanisms associated with these Western diseases with the hope that one day their knowledge that they're gaining will contribute to a drug that can treat one of these diseases that could be prevented. It's just, it seems like a crazy system to me. And so I really like this, this cartoon. Great news, Captain. You can inform the passengers we have slowed the rate of sinking. And this is really what I think is happening. So the question is, what are the root causes of these problems? How do we get in front of this? How do we figure out a situation where our kids won't get the same diseases that we're watching our parents get? And so if we look at the dozens of diseases that are going up in synchrony, it's possible that there are dozens of causes, one for each of these different diseases, or it's possible that there are a few major causes that are actually contributing to a common mechanism that's pushing us differentially towards these different diseases. And so what could these be? Well, we've heard a bit about this already today. I think diet is a major component, and I think that the microbiota may provide one part of this unifying theory as to what is pushing us towards these diseases that are largely driven by inflammation. So this is a study that was done, published in 2000, actually two studies published in 2013, looking at the microbiota of 292 people. And they did metagenomics, where they just sequenced everything in the gut microbiota of these people. And they noted that they saw a bimodal distribution in the gene number within these microbiotas. So some people were in this group here with a low gene content, and some people were in the high gene content group. Now what they found is that the low gene content group had increased adiposity, increased circulating cholesterol, increased inflammation, higher insulin resistance, the list goes on, but there was a clear association between having a low diversity microbiota and bad things going on in your body. As part of one of these studies, they did a dietary intervention. This is the dietary intervention they did. Among other things, they had less energy overall. They had low glycemic index carbohydrates and an enrichment in soluble fiber. And they saw something very interesting. They saw the high gene content group over the course of the intervention didn't really change the diversity of their microbiota, but the low gene content group did. They actually increased their diversity up to a state that was similar to the high gene content group. And along with this increased diversity, they saw improvements in a lot of these clinical measures. So again, some evidence that maybe the microbiota, diversity of the microbiota is connected to our health. Now, if we just think for a second about studies that have been done looking at dietary fiber, there's a lot of literature out there. This is a nice meta-analysis. And they see that they combine 17 prospective studies, a total of 67,000 deaths, and this is one of the major conclusions. A 10% reduction in the risk per each 10 grams a day increase in dietary fiber intake. And you think about this study in the context of that last slide that I show you, and you can start to connect some dots, I think. Okay, so this is a timeline of human evolution since we became human, and thinking about what Rob was telling us about this morning. What have we been eating? What have we evolved to eat? And for the largest portion of this time, we were foraging as hunter-gatherers for 190,000 years of this timeline. And it's really only the past 10,000 years that we've been farming, and then even more recently, we've been pushed towards industrial processed food. And at the same time, dietary fiber is going down. It's plummeting. On average, we eat around 15 grams of dietary fiber per day. This is the nutrient that feeds the gut microbiota, that dense microbial community that's controlling so much of our biology. That's on the order of a 10-fold decrease in the dietary fiber that we evolved to eat. So these microbes in our gut are essentially starving in the Western world. And so wouldn't it be nice to be able to go back and sample the microbiota at different points in human evolution and say, what did the microbiota look like? What microbiota did we evolve with? Now, there was probably no single microbiota, but you might be able to figure out, if you surveyed enough places on the planet, some commonalities that could be rules for understanding what microbiota we evolved with, what microbiota our human genome came to understand to interact with. Well, we can't go back in time, but we can go to traditional populations and survey their gut microbiota and try to infer what an ancient microbiota must have looked like. And so there are actually 10. There was just another study published with 2 more, and it shows the same exact trends as the other 8 that have been published. I'll show you 8 populations of humans whose microbiota have been studied that live traditional lifestyles, either hunter-gatherer or rural agrarian lifestyles. And in all cases, their gut microbiota is much more diverse than the microbiota of Westerners. So this is a study of, this was actually the first one that came out in 2010, looking at people in Burkina Faso, showing that they consume way more fiber, and they have many more operational taxonomic units, OTUs, or species, bacterial species in their gut microbiota, Burkina Faso, versus the European Union, a Western cohort that they used as a control. This is a study that looked at populations in both Africa and South America compared to Americans. Both of these traditional populations lived rural agrarian lifestyle. And again, they see many more species in the gut microbiota in these traditional populations compared to people living in the United States. These are hunter-gatherers from Tanzania, the Hadza. Again, much more diversity in their gut microbiota compared to Westerners. These are let's see, this one is, these are the Peruvian, so there's one hunter-gatherer, the Matsis and the Tunapuco, who are the rural agriculturalists, again, many more observed species compared to people in Norman, Oklahoma that served as the controls. Papua New Guinea, same story. Uncontacted Amerindians in the Amazon, same exact thing. Way more diversity than people living in the United States. So what could be going on here? Why do we have less diversity in our gut microbiota? Well, there could be many things that explain this. We're eating a very different diet. We live in a sanitized environment. We take antibiotics, which kill microbes. And then we have other medical practices that probably limit microbial transmission between generations, things like C-sections. And so we're particularly interested in diet. We think that this is probably the really important factor out of this. So this is a painting by Banksy. I think it captures the sentiment of the slide very nicely, and it's really the idea that there's, could we now have, all of us living in this room, an incompatibility between our microbiota and our human genome? We've evolved over a long period of time with a microbiome that appears to be relatively stable. If you look at these traditional populations, they have many commonalities. Many of those populations have been separated by tens of thousands of years. So it suggests that we had a very stable microbiome that probably had, perhaps not optimized, but better interactions than we have now with our gut microbiota. But our gut microbiome is incredibly malleable. It changes really rapidly with diet, and it can change much more quickly over the evolutionary time than our human genome. And so as you change your diet, you essentially are changing this entity, and it no longer interacts nicely with the human genome. So you have this asymmetric plasticity between these two entities that need to play nicely together. So we wanted to figure out if it was just reduced diversity. Do we have the same species as these traditional populations, or are we actually missing groups of bacteria as a collective group, Americans in general? And so my graduate student, Sam Smits, took the data that was the portion of the traditional data that was compatible with the American data that we had, and put it all on a single plot, looking at all the bacterial species in the gut microbiota from hunter-gatherers, rural agrarians, and then people in the United States. You can just focus on the bottom black line for each one of these. That shows the presence and absence of a given bacterial species. So if you see a little black line, that means that bacterial species is present. And so you can see some immediate trends here. There's sharing of species in the people that lead hunter-gatherer lifestyles, rural agrarian lifestyles, regardless of whether they're in Africa or in South America. And these are species that were completely lacking. This is from over 200 people in the United States. So huge groups of bacteria that we likely co-evolved with are completely missing from the American microbiota. What does this mean for our health? And so, I work closely with my wife, Erica. She's a scientist in the lab, and we wrote this piece together in Cell Metabolism, Starving Our Microbial Self, where we postulate that there's been this reduction in diversity of the gut microbiota over time due to this change in diet. And if you don't want to look up this reference, we actually had a op-ed piece in the LA Times just 2 days ago where that distills a lot of these ideas down in a simpler format. And so we wanted, so this is a model, a hypothesis. We wanted to test some of this. It's tough to test multi-generational experiments in humans, and so we turned to a mouse model to do this. And so the first experiment that we did was to take mice and give them a human microbiota so that we could study human relevant microbes. We gave these mice a high-fiber diet, and then we split the mouse group into two separate subgroups, one that continued to receive a high-fiber diet and one that received a low-fiber diet. The expectation was that diversity would crash in this group, and then the question was, once that diversity crashed, would it come back, or had it been lost irreversibly? And so this is data from that experiment. So here we're showing the change in the abundance of different species as they're transferred from the high MAC to the low MAC, or high fiber to low fiber diet. We see this tremendous reduction in a large number of species on the low MAC diet. We take those mice, we put them back on the high MAC diet, and we see that a lot of these species actually come back. They bloom again. So this is the property of resilience in the gut microbiota, that there's malleability, but it can actually come back after it's been perturbed. But there are a group of bacteria here that don't come back. And so this is indicative of scarring, of something persistent that's ongoing after the perturbation has stopped. Okay, so that was one generation. We were curious what would happen if we did this over multiple generations. What if we took mice, we put them on a low-fiber diet, and then we started to breed them? And then we took those mice and we bred them and took those pups and kept them on a low-fiber diet the whole time. And then after we wean the pups, we then take the parents and we put them back on a high-fiber diet and ask the question of what can come back in each successive generation. So this is these are the results of that experiment. So here, stick with me for a second. This is a little busy. Each one of these columns represents a different bacterial species in different generations, 1, 2, 3, and 4. How abundant it is reflected in the color. So for instance, this group of species here are rock stable over the course of the entire experiment. Over here we have species that are more variable. Each column here is an individual mouse, so we had 5 mice per generation at 2 different time points. And so we can see that there are some that are more variable and actually some species that are actually lost. This is on the high fiber diet. We're actually seeing some loss of microbial species, but not many. Overall, the diversity of the microbiota is maintained. What about the group that received the low fiber or low MAC diet over the course of the 4 generations? The microbiota just degrades, it deteriorates. So you see that in generation 1, you get this loss. Some of these species that are lost actually come back when you put the mice back on a high fiber diet, but they aren't passed on. And that happens here as well. You have species that you don't see, you wean the pups, you put the parents back on high fiber, they come back, but then they don't get passed on. And so what we're seeing here is diet-induced extinction in the gut microbiota that's compounding over generations. And this is maybe what's happening in our culture right now. And so to bring this back to human data, that's all mouse data that I've shown you. I presented these as different modern populations of humans. These are all extant populations, but perhaps we can use them as proxies of different times in the past. Hunter-gatherer, rural agrarian, modern. Looks kind of similar. So a major question is this diversity important? Are these lost species important? We know that if we lose cell types associated with our human part of our biology, that's really important, right? You lose beta islet cells or you lose myelin, that's meaningful. Potentially losing specific bacteria that are performing specific functions for us might be very important. So is this diversity important? And if it is important, how do we get it back? Can we use diet alone? Do we need to reintroduce the microbes that have been lost? And so these mice here on generation 4 with the lower diversity are actually being fed a high fiber diet and their diversity isn't coming back. So we thought, well, let's reintroduce the microbes and see what happens. So we performed a fecal transplant between the control group and the low diversity mice. And we see that within 3 days of the fecal transplant, all the diversity comes back and it's maintained. So we may have to reintroduce microbes if these turn out to be important, the microbes that we've lost. So reintroduction of missing microbes rewilds the gut microbiota, and microbiota reprogramming may need to involve strategies that incorporate changes in diet, dietary MACs, as well as taxa that are not currently present in our Western gut. And so this study that I just showed you data from was published in Nature last month, Erica is the lead author of it, and it really begs this question of how important is diversity, what diversity is important, and how do we regain it if we need to. This is one of our collaborators in the field that was mentioned earlier today. Rob mentioned Jeff Leach. So Jeff, some of you may follow Jeff's blog. He is doing incredible field research, sending us hundreds of fecal samples from the Hadza. But he also has performed a fecal transplant on himself in the field, as many of you know. He's described this in vivid detail on his blog. And so we're actually analyzing his microbiota now, and we'll have results from this at some point soon. But the question is, can he get microbes from the Hadza to entrench in his Western microbiota? How abundant do they become? How long do they last when he comes back to the United States? So we'll have answers to these questions at some point soon. But so this is the general model, simple model. Dietary fiber leads to increased microbiota diversity and metabolic output and protection from Western diseases. So why protection from Western diseases? We know that most of these diseases, if you look at cancer, heart disease, autoimmune diseases, obesity, metabolic syndrome, all of these diseases are a result of inflammation. They're all driven by decades of simmering inflammation. And so it could just be that we're operating at the wrong inflammatory set point, right? So look at what happens to fiber when you eat it. So we have two different metabolic scenarios here. This is a digestive tract. The stomach would be up here. Food passes through. If you're eating a high fiber diet, you're feeding complex carbohydrates to your gut microbes. They produce these short chain fatty acids that go into your circulation. And at least in mouse models, these have been shown to have a variety of regulatory effects, one of which is many different tolerogenic effects on the immune system, inducing Tregs, recruiting different dendritic cells to the lung that are less reactive. So if these same mechanisms are operating in humans, it suggests that just eating more dietary fiber, producing more short chain fatty acids can quiet your immune system. And imagine over decades what impact this could have on Western diseases. Now this is what happens if you eat a Western diet. You consume a lot of simple carbs and fats. Those get broken down and taken up into your circulation in the upper GI tract. You end up with high blood sugar instead of short-chain fatty acids. Very different metabolic scenarios in your circulation. And then the other thing that happens is the gut microbes, because they're starving, they turn to the only source of carbohydrate that they have in the gut. And guess what that is?
Mucus.
It's green. Mucus. Mucus is made of carbohydrate. It's carbohydrate on protein, but it's primarily carbohydrate. And the bacteria in our gut are really adept at using it because it's a great fallback source of carbohydrate, consistent endogenous source of nutrition if times get tough. So, one project in the lab that I started with was imaging the gut. We thought that a major gap in just basic knowledge about the gut microbiota was understanding how the microbiota is organized spatially. We just had a paper come out in Cell Hosted Microbe last year called Quantitative Imaging of Gut Microbiota Spatial Organization, a paper that I'm really proud of, a collaboration with a bioengineering group, Casey Wong's lab at Stanford. But the importance of spatial information, I think, is nicely illustrated by this Swiss photographer and comic, and he likes to organize things. And so here he's taken a fir branch and he's organized it into the different components. So needles, branches. This is similar to conducting a 16S ribosomal enumeration, a census of the gut microbiota. We know what's there. But it's so much more informative to know how these things are organized, right? And so that's really what this project was driving at. And so this is one of the key findings of that study that we presented last year, that mucus thickness in humanized mice decreases if you're not eating dietary fiber. And so these are mice that are given a human microbiota and then fed either a fiber-rich diet, a fiber-deficient diet. Host tissues on the left, microbes are on the right. Yellow is plant material going through, feeding the microbes, and green is mucus. So just focus on the green. That's the fence. Good fences make good neighbors. This is true in the gut microbiota. We know that this is a nice, thick mucus layer here. Look at what happens when you're on a fiber-deficient diet. The mucus layer thins, the microbes get closer to host tissue, and we know from surveying host responses that they start to incite a simmering level of inflammation on the host side. Now this was only done over the course of weeks in a laboratory. Imagine what happens in a Western gut over the course of decades if your microbes are relying upon your mucus layer instead of dietary fiber. This is just, as I mentioned, it was quantitative imaging, so these are histograms of the mucus thickness to show you when you're doing those sorts of microscopic studies and you zoom into tissue, you have this huge section of tissue and you zoom into one little part, you can see pretty much anything you want depending upon different areas that you go to. And so what we did is we developed a method that could survey the entire length of tissue and gather all that data in an automated way and then present that data in a histogram. And so this is around the length of colon for a high fiber diet versus a no fiber diet, and you can see that there's a huge difference in the mucus thickness here. Okay, so I wanted to transfer really quickly to just where the field is headed. We're, as I mentioned, we're not just looking to the past, we're also looking to the future in this field, and I think one of the futures is engineering gut bacteria to conduct specific tasks within the gut. I think we can probably address a lot of the problems, 80–90%, just through diet if we start eating the right diet. There will be problems left over that we need to deal with in biomedicine. And we're hoping to engineer gut bugs that can sense inflammation and secrete an anti-inflammatory protein as they detect it. So you get site-specific, dose-specific delivery of anti-inflammatory that will turn itself off when the inflammation goes away. You can also get microbes to perform diagnostics, and it's starting to think about the gut microbiota as a technology platform, not just part of our biology. This is using that project. We've actually gotten different bugs to glow, actually express different fluorescent proteins. So this is 6 different species of bacteria, all living within plant material within the gut. You can see there's different microcolonies of the different species. Each one has a different fluorescent signature, so we can distinguish them. And then these are mice that are developing colitis and then treated with an anti-inflammatory protein. So these are controls that are losing weight, and these are 2 groups that are receiving anti-inflammatory peptides of 2 different flavors, and they lose weight less rapidly and have less severe inflammation. So we're on our way to doing this. And then another really important part is understanding that the gut microbiota is secreting all sorts of interesting small molecules in our gut. They're like a little unsupervised drug factory. And these microbes are different in each one of us. They're secreting slightly different molecules. These molecules are making their way into the circulation. They come from things like, aromatic amino acids like tryptophan and tyrosine. You get things like indoles and cresol that get taken up, co-metabolized by the liver. We have a very kind of beginner idea of what these molecules are and how they interact with our biology. There's hundreds of them. We have to define what they are and how they're interacting with our bodies, how to optimize these, perhaps optimize them in a personalized way so that they're not causing disease and they're lending to our health. And so this field is really moving towards, because we're moving towards precision or personalized medicine, and the microbiota is such a huge lever on human biology, it's so malleable, and it is a major determinant of individuality. It's going to be a major component of personalized medicine. There's no doubt about it. And so we're going to end up with microbiota typing as part of going to the clinic, perhaps microbiota targeted treatments, because the microbiota is such a great lever on so many different aspects of our biology. But so the question is, because all of us have different microbiotas, and there are hundreds of species in there that change over time, day to day with each meal, what is the horizon for doing something like this? How long is that going to take? And it's probably going to take a while, and so what can we do in the meantime? Can there be a simple solution to some of these complex problems? And I think this comes back to diet and some really simple things that you can do. So this is an image from a New York Magazine article. They profiled our family, and part of this was because we wrote a book, and in this book we document how we've just changed the way we live because of this basic research that we're doing. We noticed that we were changing the way we lived, we were changing drastically what we ate. We saw that other scientists that didn't study the microbiota were not doing this. Other friends that were non-scientists were not doing this. Other scientists that study the microbiota convergently were doing the same exact things that we were doing. And we thought, well, this just isn't right. We have a privilege to information. We have privileged access to information that should be accessible to everybody. Everybody should be able to know about this and then make a decision about how they live their lives. And so that was really the purpose of us writing our book. The author notes that we're making a commitment to fiber consumption that borders on the comical. And so, that's one view of it. We moved from probably around 15 grams per day up to around probably 37, what's recommended. This is what the Hadza eat. So what is really comical here? Is it the fact that we've gone here, or is it the fact that there's such a huge gap here? So we really have to think about what the right context is. And so just a personal story. So just over the past 5 to 7 years, as I said, we've drastically changed things, started gardening, eating a lot of fresh vegetables from our garden. This is compost in our yard that my daughters like to play in. They love pizza, and I'll serve them pizza, but this is my style of pizza. So this is a sourdough crust made with flour that I actually grind from wheat berries, so into a very coarse flour, so it's dense and it's not very refined. On it are fava beans, onions, some olives. So that's the type of pizza that we should be feeding our gut microbiota. I also make my own kefir. Every day I have a small glass of kefir, a way to introduce new diversity to your gut microbiota. And then we also got a dog. I mean, we wanted a dog anyway, but dogs are, you know, contact with nature, contact with soil, contact with animals like this is a great way to expose yourself to a lot of the microbial diversity that we're missing in our lives. And yes, he's actually as mangy as that picture shows. His name's Louis, for Louis Pasteur, of course. And this is what's happened to my gut microbiota over the past 5 years. So these are the number of species. I started off at around 700 to 800, and just last year I surveyed it. I'm up around 1,400 species. So this provides some hope, I think. I probably still haven't acquired these critical groups that were, or potentially critical groups that I'm missing, but I have increased overall diversity. Now, this is an N=1 experiment. I can't attribute any of this to this. So we've actually started a Center for Human Microbiome Studies recently at Stanford. We're going to start large-scale human studies to try to do this in a documented way, very controlled way, and try to figure this out, connect it to human immunology with Mark Davis. We're working with Christopher Gardner there to do the dietary interventions, something that we're very excited about. And then I just want to finish up by thanking my group. I have a great group of scientists that I work with at Stanford, and a lot of the ideas and data that I've presented today are a result of them. I just get to come here and tell you about it. Check out our website at Stanford and our Facebook page. My wife documents a lot of the research that's going on in the field. And then I also want to thank funding sources, the NIH, NIDDK, and the Burroughs Wellcome Fund. And then thanks to all of you for your attention.